Okay, great. And let's see. Looks like I'm going to go ahead and share my screen just real quick to run through an introduction. So hopefully everybody can see the Chelsea's Hope logo research webinar. Is that correct?
Yeah, looks good, Frank.
Great.
Yeah, Frank, it looks good.
Okay, great. So what we're going to do today is we're going to run through some quick introductions. And then Professor Matt Gentry at University of Kentucky College of Medicine is going to talk about concepts and data for the VAL 1221 candidate. And then we're going to have a roundtable discussion with Rob Shaffer and Dustin Armstrong, who are co-founders. Oh, what happened to your PhD, Dustin? Apparently they pulled your back. Somebody didn't like your dissertation there, buddy, and they just pulled it back. You might want to get in touch with let's see, you were Illinois, Chicago, right?
Correct.
So you need to reach out to them and let them know that that your data is actually legitimate, and tell them to give those 3 initials back. And then we'll have an audience Q&A. Actually, I'll leave it to the 3 presenters to talk about how they want to do that. I don't know what happened with my red line there, but as you were having some issues. So real quick background for our presenters here. Matt Gentry, many of you are familiar with Matt. He's a professor of molecular and cellular biochemistry, University of Kentucky and College of Medicine. He's a very good master's committee member, as I can tell you from a personal experience, as he was on my daughter's master's committee there. He's director of the Lafora Epilepsy Cure Initiative and director of the UK Brain and Epilepsy Alliance for Metabolism Research. Bachelor's at the University of Evansville, doctorate at Syracuse, and did his postdoc with Jack Dixon at UC San Diego, and that creates a real close alignment with Chelsea's Hope, because Jack Dixon is related through marriage with one of our families. It's actually Kim Rice and Matt wrote a piece describing how this happened, and it's on the Chelsea Hope website. I encourage you all to read it if you haven't. We have Rob Shaffer, who's co-founder of EnAble Therapeutics. I'm not even going to try Rensselaer Polytechnic Institute for his bachelor's, and Boston University Biochemistry. Rob, I did not list Framingham High. And then Dustin Armstrong, another co-founder of EnAble Therapeutics, University of Chicago, University of Illinois, Chicago, and then did his postdoc at Novartis. So that's it for our presenters. Let's go ahead and give them all the time. And so I'm going to go ahead and stop sharing, because you might want to share. And let's go ahead, Matt.
Great. Thank you, Frank, and good to see everyone. So Dustin, I'll go ahead and just run through some data slides, and then we'll launch after that. Does that sound good?
Sounds great. Yeah.
Okay. So you all see the correct window there?
Yes.
Okay, great.
Yes.
Okay, so what I'm going to do is go through sort of the data with the antibody enzyme fusion platform that's used by, you hear me probably refer to a few different companies, and Dustin can answer questions about this afterwards. Started out as Valerian Therapeutics. The patent and intellectual property is held by Parasail Therapeutics, and the company that he and Rob have co-founded is EnAble. So if I use their Valerian, Parasail, EnAble, think of them as one entity, although they're not one entity, but think of that way for these slides. So Lafora disease, which you're all very familiar with, just as a basis here, this fatal autosomal recessive horrendous disorder, patients developing normally for the first decade of their life, experiencing an epileptic episode, that progressing over the course of the next decade through myoclonic events, which you see in this beagle dog here that has Lafora disease, also accompanying massive dementia aspect of the disease, which mimics what some are starting to refer to as sort of a childhood Alzheimer's disease. Hallmark of the disease are these sugar-like aggregates found in cells from most all tissues, including the brain, called Lafora bodies, which are shown in stained in dark pink here. We in the field have shown, many labs have shown that these are the driver of the disease. And so a lot of work has coalesced around removing or inhibiting their production. So just in terms of where we stand as a field from the last 100 years or so, the disease was described in 1911 by Gonzalo Rodriguez Lafora. In the late 90s and early 2000s, 2 different genetic loci were identified that cause the disease. So recessive mutations in either the EPM2A gene that encodes laforin or the EPM2B gene that encodes malin result in the disease. Over the course of about the next 15 or 20 years, many groups in the field, including my lab, have developed cell culture models, biochemical assays, and mouse models to really figure out what are these 2 proteins doing and how is it that when those proteins are mutated, how does that drive the disease? And so using the foundational science that's been developed in a number of labs around the world, in 2016, we received a large grant from the National Institutes of Health to really focus on transitioning these foundational discoveries into preclinical therapeutics. And I'll talk a little bit about one of those here today. I've got listed all of them here, and in a couple of slides, list them again as well. So this is a slide just capturing the basic mechanism of the disease. We know quite a bit about what the protein laforin and malin are doing. That's not exactly germane to what this discussion is about, but we have a good foundational understanding of what these proteins do. And we know that when they're aberrant, that leads then to aberrant glycogen metabolism. That aberrant glycogen metabolism results in a Lafora body. That Lafora body drives the disease. And so the Lafora disease research communities developed a number of different mouse models that do a good job of mimicking the disease. You see here a brain slice from a wild type mouse. This is looking at a Lafora knockout mouse and a malin knockout mouse, and you see those dark pink Lafora bodies in both cases. These mouse models exhibit neuroinflammation, they exhibit neurodegeneration and epilepsy, just as what is seen in the patients. We also have an antibody against Lafora bodies, and the point of this slide is just to really highlight the fact that these Lafora bodies impact every region of the brain. Here's the hippocampus, the cortex, cerebellum. So when we think about a therapeutic, we have to think about a therapeutic that impacts every region of the brain. And that's going to be important as we start moving from the preclinical phase into the clinical phase. That clinical drug has to reach every portion of the brain. Okay, so the research community has really coalesced around this idea that it is the Lafora body that's driving the disease. And so the goal is to either stop the synthesis of the Lafora body, or as you'll see today, actually degrade those Lafora bodies. So this is a simplistic way of how we think about the disease. In a healthy brain, you've got glycogen synthesis occurring. That glycogen builds up to a certain level. There's also glycogen breakdown, and that glycogen breakdown results in glucose being released that drives normal cellular metabolism in the brain. In Lafora's disease, patients can generate a glycogen-like aggregate, that Lafora body, but they can't break it down. And so you get aberrant metabolism. And so a number of labs have shown that by inhibiting that glycogen synthase gene, that GYS gene here, if you inhibit it 50% or more, that's enough to sort of let cellular balance come back into play, and you see then normalized metabolism, decrease in inflammation, decrease in neurodegeneration, and decrease in the epilepsy. So this genetic method that was shown here using the Lafora disease mouse models really set the storyline for developing a therapeutic option, and that therapeutic option targets either inhibiting glycogen synthesis or degrading those Lafora bodies. And that was the basis of the NIH grant that we received back in 2016. The title of that grant is Lafora Epilepsy. We wanted to continue discovering the basic mechanisms so that we fully understand the disease, but also start to transition those into preclinical therapeutics. You see there all of the different sites that are associated with this grant across the world. Many of you have heard our talks at the annual meetings. And again, the basis of this grant is summarized here, that it's the Lafora body that's driving the disease. So if we can remove the aggregate, we can either stop progression or potentially even reverse progression of the disease. So this is sort of a snapshot showing where the field is at the moment with respect to therapeutics. There is an antisense oligonucleotide that many of you I'm sure have heard about from Ionis Therapeutics that was developed in collaboration with Bergman Ashen's lab. It's been announced that that would go into the clinic in the second quarter of 2022. There's an antibody enzyme fusion that we'll talk about today. There's the small molecules that are being developed at Indiana University. Repurposing drugs that really Pascual Sands and Jose Ceratosa have worked on, and then there's some entry-level gene therapy being developed in some labs and in collaboration with companies. But again, the idea for each of these is to either stop or ablate these Lafora bodies. So what I'll tell you about today is work that we performed in collaboration with Dustin and Rob, Valerian Therapeutics, and EnAble. This antibody enzyme fusion that has been administered into mice via intrathecal administration, directly into the brain via intracerebral ventricular administration, and then also into mice systemically just via intravenous administration. Okay, so the Parasail and EnAble and Valerian platform uses this idea of what's called an antibody drug conjugate. This is quite popular in the cancer field right now. There's hundreds of clinical trials going on with antibody drug conjugates for different types of cancer. And the idea is that you can generate an antibody that will target a specific cancer cell type, and you attach then to that antibody a cytotoxic drug. So the antibody is a delivery mechanism to take this cytotoxic payload to the cancer and then kill the cancer. Well, Parasail, or Valerian and EnAble, uses a different version of this type of technology. They use what's called an antibody enzyme fusion platform. What they've identified is a fragment of an antibody. That fragment of an antibody is called a Fab, and the real power of this Fab is that it can gain access into a cell via this ENT2 transporter. Once inside the cell, it has access to both the cytoplasm as well as the nucleus. And so what we did in collaboration with Valerian was identify an enzyme that can degrade Lafora bodies. This is pancreatic alpha amylase. We all have amylases produced in our digestive system that can degrade carbohydrates. And so this is one of those degradation enzymes. So in collaboration with Valerian, we fused their Fab with this pancreatic alpha amylase. This fusion protein is referred to as the drug VAL-0417. And I'll also, at the very end here, talk about another drug, VAL-1221, we've got data on both drugs that are very similar. So both drugs can enter cells. Once they get into cells, the idea is then that they can degrade those Lafora bodies and release that glucose sugar to normalize metabolism in the brain. So in order to test these drugs, first we purified Lafora bodies from the mouse model. So this is Lafora bodies from a Lafora disease mouse brain, heart, and skeletal muscle. We took those Lafora bodies and then incubated them with the valerian drug. And what you see is with no treatment, the Lafora bodies are intact. You treat with the drug in a test tube and it just explodes those Lafora bodies and degrades them. We can also do this from a tissue slide. So this is just looking on a slide. We can stain for those Lafora bodies. This is from the Lafora disease animals again. You can see those Lafora bodies with no treatment. If we just treat with buffer, and then if we treat with the valerian drug, you see just the empty holes here where the Lafora bodies were. They've been degraded. So that's all great, but more importantly, you want to see, does the drug work inside the tissue of the animal? So to test this, we at first did intravenous injections of the drug. We used quite old Lafora disease animals. These animals have robust Lafora bodies. We did 4 injections over the course of 2 weeks and then euthanized the animals and then looked at the Lafora body loads in multiple tissues. And so this is just showing Lafora body loads in the skeletal muscle, but we also looked in the hearts of these animals. And what you see here biochemically is that when we treat the Lafora disease animals with the drug, we very much ablate those Lafora bodies. You can look at this visually as well. In this particular slide, the Lafora bodies are these larger dark purple aggregates. And you see that with PBS treatment, there's a lot of those aggregates we treat with the valerian drug, and there's very few aggregates that remain after just a 2-week treatment, 4 injections into these animals. So while this was exciting, really the key experiment here was, can we get the drug into the brain? And once we get it into the brain, does it penetrate into the brain enough to degrade the Lafora bodies? So to test this, we did an experiment to see, could we get the drug in via intrathecal administration? This is into the spinal column, as well as testing that against intracerebroventricular administration. This is into the ventricular space of the brain. So this is a direct infusion into the brain, which is used in some treatments of neurocentric diseases currently. Not this drug, but other drugs are administered via this mechanism. And so what we found was that both administrations gave us very good biodistribution throughout the entire brain. So we sectioned the brain into 6 sections, and then we just asked, could we detect the drug in the brain? And so this is via intrathecal administration. We could see the drug in every region of the brain, and then we could see more of the drug if we did the ICV administration. So in the mouse model, at least, ICV yields more drug into the brain, but both methods, I would say, gave very promising results regarding brain distribution of the drug throughout the brain. Now, this is an important slide here, because what we're testing is, what if we just take that amylase without the valerian antibody fragment attached to it and inject it into the animals versus the valerian drug? And what you're looking for here is brown staining. And so what you can see quite easily, just with the eye, looking at some microscopy of these tissues, is that in every region of the brain, so the cortex, the choroid plexus, the medial cortex, and the cerebellum, there's much more of that brown staining with the valerian drug versus just the amylase. So what this is telling us is that when we inject the amylase into these animals, it doesn't penetrate into the brain. It instead likely just stays in the CSF, the cerebral spinal fluid, and then eventually gets degraded. Alternatively, the valerian drug penetrates deep into the brain and enters the cells within the brain, which is a key component of it being able to degrade the Lafora bodies. So really the key experiment here was, okay, we know that the valerian drugs can degrade Lafora bodies. We know it's active in tissues. We know that it can get into the cells in the brain. What happens once we administer the drug into Lafora disease animals and then look at Lafora body loads? So we did this with a continuous infusion of the drug for 7 days, euthanized on day 8, and then again looked at that staining to see what Lafora bodies looked like. And this is where we were extremely excited about the results. You see the untreated animals have that purplish staining for the Lafora bodies. This is the negative control, the PBS treatment without the drug. When we administer the drug, we see no Lafora bodies present, and this tissue now looks very similar to the wild type tissue. And importantly, this occurred in every region of the brain. So here's looking at the cortex region of the brain, this part right here. You see Lafora bodies in the control. They're completely gone in the valerian treated. The thalamus has a lot of Lafora bodies. Again, they're gone. The cerebellum, that hindbrain portion right over here. Again, lots of Lafora bodies in the control. They're gone after treatment, and the brainstem as well. So in every region of the brain that has Lafora bodies, the drug was able to penetrate into that region, get into the cells, and then degrade those Lafora bodies. So that was a spectacular result. Then though, we wanted to know what happens with respect to the brain and its physiology once those Lafora bodies are gone. Now, the Lafora disease mouse models, they do exhibit neurodegeneration, and they do exhibit some aspects of an epileptic phenotype. But as a field, we know that those are not the strongest of phenotypes. And so what you would like to see as a readout, a biomarker, is a very strong and robust phenotype. And so we talked to a number of industry experts about how could we come up with a new biomarker to show that a surrogate for the activity of the valerian drugs, to show that it was being effective. And the advice that we got was to turn to this workflow of using metabolomics. And so the idea here from metabolomics is that you can take any tissue type that you've extracted from a euthanized animal, and you can pulverize that tissue down to a very tiny powder form, almost like flour, baking powder type extract. And then once you have those particles, you can extract from them the polar metabolites from that tissue. And what those polar metabolites are metabolites from central carbon metabolism. So these are metabolites from the TCA cycle, glycolysis, and the pentose phosphate pathway. So these are, in essence, the molecules in the brain that tell the brain what to do. It allows neurons to fire appropriately. It allows for the sleep-wake cycle, for thought, for memory, everything that allows us to be who we are. These metabolites drive those processes. And so once we have those metabolites in solution here, then we can do some analyses on them using what's called gas chromatography mass spectrometry. We can then curate the data and then do some mathematics to look at how close the diseased animal metabolic profile is to the control animal. And so what I'm going to show you is one of these principal component plots like you see here. And each dot within the principal component plot measures each one of these metabolites. So we measure about 100 or so metabolites for each animal. This is a list of some of those metabolites. So we take all of these data and we boil them down to one dot. So this is a very data-rich plot here. And what you're looking at is the brain metabolic profile of a Lafora disease animal versus a wild type animal. So this is important because we're seeing very good clustering of the wild type animals from the knockout animals. So this is that good phenotype that very robust phenotype that we're wanting to see. So the question then is, if we treat these animals with the valerian drug, do they look like the wild type animals now? And so first we do the control. We just take these animals and we treat them with PBS. Treating them with PBS means that we're doing brain surgery on these animals, which increases inflammation and changes their metabolic profile. So you see that metabolic profile shifts here. We then take Lafora disease animals, and we treated them with VALO-417. And to our delight, these animals now look exactly like the wild type animals. So we've not only degraded the Lafora bodies, but we've normalized the brain metabolism of these animals. There's an additional way that you can do this. You can take all of these data, throw it into an algorithm, and ask the computer to say which groups of animals look most like each other. And what the algorithm comes out with, it can say, okay, these are all of the Lafora disease untreated animals. They all look the same, and they cluster right here. Over here in this cluster are the wild type animals and the Lafora disease animals treated with VALO-417. And importantly, the algorithm can't tell the difference between these animals. So the diseased animals that have been treated, their brain metabolism looks just like the wild type animals here. So that's a very reassuring and exciting result regarding what this drug is doing to the metabolic profile of the diseased animals. Now, most recently, just in the past week or so, we recently published an additional phenotype of the Lafora disease animals, and this involves a process called glycosylation. And the process itself isn't important, but what is important is that in wild type animals, you have these sugar decorations that are depicted by these schematics here. And so what these data are showing is where exactly is this sugar located in the brain. So it's in this part of the brain. And then this tells you where are the high and low levels of this particular sugar. You see this sugar has a different distribution, and now this here sugar has even a different distribution for itself. So this is in the wild type brain. In the Lafora disease animals, we see this massive hypoglycosylation defect. So there's decreased sugar in each of these regions of the brain for each of these three types of sugars. So the question then was, what happens when we treat the Lafora disease animals with the valerian drug? And excitingly, we rescue each one of these phenotypes for these sugars. So we've now shown that we rescue both the metabolic profile and the signaling profiles in the Lafora disease animals whenever we treat with the valerian drug. So just in conclusion then, what we know is, and I haven't shown all of the data, but we have data for both the Malin knockout animals and the Laforan knockout animals with VALO-417. Both animals respond equally well with the drug. So when you think about this in terms of patients that have mutations in Malin or Laforan should be candidates for treatment once this is transitioned from the preclinical phase to the clinical phase. We're currently working to define the minimum dose with maximal response in the animals. We're trying to define what's the minimum length of time for treatment. Right now, the least amount of time that we've treated an animal is 3 days to see that very robust response with the drug. And we're also working to establish human and mouse biomarkers for the drug. Now, when Frank introduced this, he talked about VAL-1221. This is a very similar drug that valerian has. It uses that same antibody fragment as what's found in VALO-417, but it has a different enzyme attached to it. So whereas this is the pancreatic alpha amylase enzyme, this is an enzyme referred to as GAA. They call the drug VALO-417. It's got some unique characteristics that are quite attractive. It has 2 ways to enter into cells, which allows potentially more of the drug to get into cells. And the data that we've generated from the mouse models shows that it's just as effective as VALO-417. So at least in preclinical data, there's 2 drugs that can be used to treat Lafora disease mice. From the valerian Parasail/EnAble drug portfolio. So I just want to point out, Parasail is not the only company that's utilizing this technology. This is a technology that's very hot right now and on the cutting edge of research. Denali Therapeutics, Roche, Ionis, Merck, and Pfizer are all developing programs for other types of diseases using these antibody-based platforms. So, this isn't, this is a space that's being very well developed, and major companies see it as a very rich platform to use to treat a number of different diseases. And so with that, I should acknowledge the funding that we've gotten for this work. There has been funding from Valerian or Parasail for this work, as well as NIH funding. I've collaborated with Rob and Dustin and their teams, as well as a group at Indiana University, and really Katie Brewer, a former graduate student in the lab, did a lot of the work that or maybe all of the work that I presented to you all today. So I want to acknowledge her great efforts in this as well. So with that, I can stop sharing. And I don't know, Dustin, how you want to take questions, or does that work?
Yeah, we can ask Frank what he would like to do.
Or if you want to add anything else to it, feel free to clarify anything
Yeah.
the next time.
No, you did a great job.
You and Rob can offer your clarifying comments, and then we'll start on some questions.
Okay, yeah. First, I'd just like to thank Matt. Obviously, you can tell he's a brilliant guy. His lab has been, super productive and motivated in this field, and has produced incredible work. So, just been a great collaborator, and we definitely appreciate all the work he's contributed. He's done great. I'm sorry for the many company names, and hopefully that doesn't confuse anyone. I can give you just kind of some quick background. So as he mentioned, Valerian Therapeutics was the original company that I had founded, and I was the chief scientific officer. Rob worked with me at Valerian. Unfortunately, Valerian, like some companies in the space in development, fortunately couldn't get continued funding and was forced to close operations. Following that, Rob and I now founded this new entity and are in the process, early stage now, of fundraising anew and kind of a reboot where we hope to raise additional dollars to bring these drugs back into the clinic. In the case of 1221 and newly developed the 417. So we're happy to answer questions and give status updates there. This transfer, as Matt mentioned, just happened kind of this year. And so we're very new. As you can tell, we're a very, at this stage, a small company, but we're very excited about the assets that we have. In some cases, as Matt mentioned, the 1221 has already been in patients in another disease, in Pompe disease, and it completed a phase 1/2 clinical trial, and patients were on drug up to 18 months. So we have well-established safety data with that drug, but that also bodes well for their antibody used in both drugs, that it could be safe in patients to utilize. And so that's very valuable, data. We have current drug material for 1221 already stored that could be used for a clinical trial. So we're pretty advanced in terms of having that asset there in place. And we have a significant patent portfolio. But with that comes, expenses. And so we have, funding that we need to raise that will allow us to continue paying to store the drug at really ultra cold temperature, to continue processing patents that allow us to have a viable drug in the future. And so we're, in very active stages now of looking both from venture capital funding through partnerships with other companies in any way possible that we can advance this. And that's why I'm talking today also with Chelsea's Hope and with other groups, another way to raise funds is through grant writing. And so Rob can speak more to that, but we've been very active in seeking NIH grant support, in some cases a clinical grant application where it allow us to get back into the clinic with funding through that mechanism. So we're in the very early stages. We're very, very excited. And as you can see from the data, very encouraged about the opportunity.
Yeah, great. So I guess I can share just a few comments to add on to what Dustin was saying there. For the NIH clinical trial grant that Dustin mentioned, so this is a brand new funding opportunity from the NCATS division at the NIH. And basically the goal there is to fund a small basket trial, so multiple indications in one trial that share a similar underlying cause. And so here we're looking at polyglucosan diseases that have aggregated glycogen. And Lafora is one of the two diseases that we're looking at in that trial. And we're hoping to get a score for that proposal next month. At a high level, just a couple of details there. We're going to have our Lafora site in Spain with Dr. Saratosa. And we're fortunate to have Matt as well on board to try to do some biomarker discovery work to try to see if we can identify a good biomarker to lend itself to subsequent trial design, patient stratification, et cetera. And so we're really cautiously optimistic about that grant that hopefully we'll hear back sometime soon. And then, as Dustin said, we are pursuing a few different grant avenues. Another one is through the Cure Epilepsy Foundation. There's a Catalyst Award that we're exploring right now. And so really trying to get our hands on a way to advance this program and try to get a couple of shots on goal, whether it's this 1221 lead asset or potentially 0417, potentially a different formulation of 1221 for different administration routes, and trying our best to improve our overall chance of success here.
Great. I'm happy to open it up to questions, Frank, or however you want to.
That'd be great. Actually, I'm going to use my prerogative to ask at least one or two questions. First of all, going back to Val 1221, just for clarification, Dustin, I think you addressed this, but I just want to be really clear. It's the Val 1221 product. Well, I don't want to call it a product, but that is now part of EnAble. Is that right? EnAble has the chemistry from Valerian now.
Yes. So Rob and I completed a transfer. We have all the business assets from Valerian. So the actual drugs themselves and IP rights to those drugs. So we have 100% control and ownership right of that. Yes.
Right. So
the 1221.
I'm sorry, go ahead, please. So the
I was just going to give some background into the ability to develop 1221 for Lafora. And as part of that basket trial, as Rob mentioned, we proposed using 1221 with intravenous administration. So that's an IV therapy would be injected, into the veins of individuals for more of a systemic delivery. As you saw with Matt's presentation, a lot of the work that we looked at in the brain was given either intrathecal or cerebroventricular into directly the brain. And so what we're proposing is that 1221 was previously in the clinic in Pampas disease with an IV administration. And that's what it's formulated for. And that's what we previously, when we went to the FDA, we now with Pompe have established that it's safe in patients. So we could go into a therapy in Lafora patients much quicker with the IV administered form of 1221. Have that drug kind of formulated for that purpose. We've already done all the required safety studies regarding that. So the real kind of scientific question then that addresses is, would an IV administered form in patients have all the benefits that would be clinically meaningful, especially in the brain? Would it get to the brain in significant quantity and have the efficacy there? And that's hard to model in mice or other animal species because they, as Matt mentioned, they don't have the exact same phenotype of patiently in terms of severity of epilepsy, and they're just different body mass, and biodistribution is more difficult to monitor there. But that is a mechanism that we have drug available. We filed for the grant. We believe internally that it would have efficacy and benefit to patients. And so that's where we could be the fastest into a clinical trial, would be using 1221 IV administered. As Matt mentioned, the Fab amylase, the 417, it does have other problems that would make it very attractive, both in Lafora disease and potentially other indications. And so we're also trying to develop that as I'd say an alternative and maybe an improved method. That's more work would have to be to demonstrate that, but it's something that we believe strongly in trying to push multiple opportunities forward.
Okay, well, that's exciting. I'm going to go back to clarification again, just because I know that our families really are eager to understand how things are discussed. So as EnAble moves forward, this may seem like a very simple and kind of very low level question, but I think this sort of clarity is really important. As EnAble moves forward, when it's discussing something like a Val 1221, is that how it's going to discuss it, or is it now going to get an EnAble moniker, if you will? Again, just so that people understand, I don't want them to think there are no drugs. I don't want them to think there are more than there are, just so it's very clear for them.
I can't promise that it won't get renamed and going forward, just because, as I mentioned, we're in the process of fundraising. And one aspect to that is when you're talking to groups about taking money, sometimes they have their own marketing ideas.
Yeah.
Ideas. And so everything could be, as it's developed, it could be changed or modified. But as we sit here today, the drug itself isn't changing. It is what it is. It's just really kind of a placeholder in terms of what you call it. And so sometimes, I think keeping it with the same name so people, as they get familiar, I think there's true benefit in that. That would be my goal, is to try to keep it as straightforward as possible.
That's great.
And just one other comment in general, there's probably going to be some jumble of vowels and consonants that the naming commission, I can't think of the name offhand, comes up with for these protein-based protein biologics, tend to have very complex, hard to pronounce names. And then, knock on wood, fingers crossed, if there's an eventual trade name that we come up with the drug, that would be yet another name. So at least the code, like Dustin said, we'll try to keep that the same. It depends on investors, but there may be another jumble of letters that is named at some point during the clinical development.
That's great. I'm going to ask one more question, but before I do that, I'm going to put out a call to everybody who is watching here and participating. You can raise your hand to ask a question. You can ask a question in the chat. I know we have one now. I'm going to try to facilitate this, and Sheila Barter, who's also on our board, is going to try to facilitate this. My last question before I give up my prerogative here. So you've got Val 1221, Pompe disease. It looked very good. Let's just push forward a little bit and say that all of a sudden that gets approved. Could that be prescribed off label to a Lafora patient at that point? What are your thoughts on that? Where could you imagine that going?
Yes, that can only happen, as you mentioned, once approval happens.
Right.
And at this stage, I think with 1221, we would have this the FDA, but it's, internally, we think it's likely that we'd have to do a phase 3 trial, likely to some extent, in Pompe disease. We don't have an immediate plan to file for approval based on the previous Pompe data. That was 12 patients total in that trial. Typically now, therapy, Sanofi, Genzyme, Myozyme therapy. So typically for applying for approval, we would likely have to do an additional clinical study in some form, either in that disease or there's other potential indications. We could do a new study and try to file approval. More than likely, the first Lafora study would be a new study that we would file a new IND, investigative new drug application with the FDA to initiate based, using this drug in Lafora. So it would likely be a clinical trial where we'd recruit patients versus patients just being able to as an approved drug. It likely wouldn't happen that way first. So we're happy to work with you. And, as you see, it's a lean organization. Both Rob and I are committed to trying to develop these drugs for patients as quickly as possible. We can recruit patients and fund a trial, and we are certainly willing to support that. Most likely it won't be simply off-label that physicians could just prescribe it because it's not approved yet.
Right.
Medication.
That makes perfect sense to me. Mike asked about timing on that. This is, of course, the magic question. It's an all-important, crucially important question to our families. You talked about a phase 3 process, and before it were to get approved for any application, in this case, I guess it would be Pompe first. Do you have a sense of what your anticipation is for that?
Yeah, I'll let Rob speak specifically, but as of right now, our fastest path would be if that NIH basket clinical grant gets funded. And as he mentioned, you never know the timeline, sometimes waiting on the government grants. We'll hopefully get a score and reviewed within a month or so. It's likely several months before you really know if you'll get funding. That in that grant, the plan is this trial patients at the beginning of 2022. And so what we're doing is trying to see, is there a way to speed that process? Can we get some funding that would allow us to do the work to get started even faster? We know the money's coming from the grant, but can we get a funder to supplement before it actually arrives, kind of a bridge? What really has to be done technically is we have drug stored, enough quantity of drug to do a trial. What you have to do is show the stability of that drug, since it's been stored now for a while. Before you go back and have it ready for a patient use, you'd have to prove that during the storage, it's still fully functional. And so that's a very defined kind of, they call it CMC, a commercial manufacturing process, where you would try to validate that. And then what you'd have to do is they call it fill finish, where you put that drug that's in these big kind of storage containers into the necessary syringes and vials that would be administered to the actual patient. And that fill finish. So those are the two main steps that would have to happen physically for the drug to be prepared. And then the second aspect that is more regulatory with the clinic, with the FDA, where we'd say, now we had this FDA for Pompe disease, we'd now like to initiate a Lafora disease. And so there's some regulatory expense to do that. So the two main buckets, we're looking for that grant to fund the regulatory expense. And then also on the clinical side, preparing the material, and then the last expense is the clinical sites themselves and funding for care and use and the monitoring involved. And so that is in the grant that we applied for, unfortunately would only allow a few patients. What we're hoping and trying to talk with investors about is increasing our funding to allow us to go into a larger number of patients and get there slightly faster. And that's what our real hope is.
So what kind of funding are you talking about? Ballpark, what level for this grant? If our families were to say pass the hat, how much would the hat have to have in it to make a palpable impact?
Yeah, I can let Rob speak to specifics. So the grant we requested is a little bit over $2 million, and that's between two different diseases. So that's Lafora disease and also Parkinson's white, which is a PRKAG2 deficiency in the heart. And the reason that this particular grant is for a basket trial where you look at more than one disease. If you have a drug you think could work for multiple diseases, the NIH is trying to encourage you to explore that and more. And so that's what this grant is for. We're speaking to investors, all the way up to requesting up to $18 million, which we think would be enough to do a full, potentially through approval study in Lafora disease to get enough patients where if it worked, you could file for drug approval. So that's kind of the high end, is up to 18 million. In the short term, to get into the clinic, as I mentioned, the grant, roughly $1 million would get us into a couple patients in Lafora. And I'll let Rob speak. We have budgets at the different clinical sites, but the cost per patient, do you want to speak to that a little bit, Rob, and what we anticipate would be kind of a reasonable cost?
Yeah, so for us, really, the biggest cost for this grant is using a global clinical research organization that would handle the trial management, handle some of the in-home treatments, and really help us with the regulatory side as well. And so because we're a two-person company, it's quite challenging to run a clinical trial, even if it's only for a few patients. So the $2+ million mark that Dustin mentioned there is really for the all of that initial stability and fill finish work that Dustin mentioned before. It's for all the regulatory submissions that are required before we can actually get into patients. And it's for that clinical CRO to say, we'll manage the trial for you, treat patients in their homes, especially for those who are unable to, or it would be very difficult for them to travel to the clinical site every other week. That could be very, very challenging. And really try to get the total budget that we would need to actually execute the entire trial without any surprises. The per patient dollar amount is a bit trickier to nail down because of those other costs, like to prepare the drug, the fill finish, regulatory assistance as well. I'm a little hesitant to speculate on how much it would be per patient, but that's something that we're probably going to have to do the math for, especially if we get engaged with investors who are looking to say how well powered could we make such a study, how many patients would we need, and then go to the FDA and say, you know, because this is an ultra rare disease, would it be possible to submit an approval package based on this one trial? It's possible. I don't know how likely that is, but those kinds of conversations are going to be crucial to have with investors, hopefully soon, hopefully in the near future. And just one last thought, just to round out in terms of the timeline for the grant. So the scientific review group that'll score the grant is meeting next month, in about a month from today, actually. The score for the grant will be released shortly after that. And then from that score, the NIH will then have what's called a council review group. And that council review will say all the grants that have a score better than X will likely get funded. And then from that basket, they'll pick out the ones that will definitely get funded. And so what we're hoping for is a final decision sometime towards the end of 2021. And then the project period, so to speak, will start early 2022, where we'll start getting the protein ready. We'll do the stability testing, the fill finish, meet with the clinical CRO, get all the regulatory submissions in line, so then we can have our IND accepted sometime, second quarter or mid 2022, and get the trial started as soon as possible after that.
That's great. Thank you very much, Rob. If you're following the chat line, our families are starting to ask about how they can help. Very quickly, Chelsea's Hope has been discussing this, whether we could actually effectuate a fundraising campaign that would, in essence, be more than a drop in the bucket. I mean, $2 million actually exceeds the lifetime fundraising history of Chelsea's Hope. And we're the biggest organization in the Lafora space. And so that just kind of tells you, in terms of the nonprofit donation experience, Lafora is very small. Now, the LECI has been enormously successful in getting research grants and will continue to be. But insofar as channeling funding, Chelsea's Hope is perfectly legally equipped and authorized to provide funding to any organization that is working in a way that's consistent with our mission, whether the organization is a nonprofit or whether the organization is a for-profit firm. We can do that legally. We have, you're talking about a fair amount of money, as I just mentioned, but our internal dialogue continues. And what you're seeing on chat is people who are asking about how they can help. You mentioned going to the FDA and the regulatory work. I know that many of our families have asked, can we write letters? Can we support? I mean, this is actually an area that I work in professionally in my paid job as a lobbyist. And I know that these advocacy plans are difficult and it's very important to get them right. And so what would you say to our families who would be willing to reach out to the FDA, get that word out, and support you with the regulatory agencies?
Yeah, we would obviously, appreciate all that support. And I think that's, like we're doing today, we want to keep the dialogue open. And I think, one benefit to being small, as you see with like Rob and I, we are the decision makers. We would be engaged. And so with some, bigger companies, it may be harder to have a personal connection or contact the right people. We want to stay engaged with the community. We want to develop this as quickly as possible and utilizes all the resources that we can. And so I think, through your group, we're happy to keep the communication ongoing. Like you said, we don't have all the expertise in all areas of drug development. We do have significant experience, but we're happy to utilize any people's talent and resources. As you said, we will probably utilize contractors and different regulatory groups, and we'll be engaged with those stakeholders in terms of, all right, are there people that we can hire internally that would develop something faster? As you mentioned, once we do go to the FDA, letters of support are very valuable. So if we're in communication, we can tell you in terms of timing when to do that and what's exactly needed. And so we don't want to leave any doors open, and we definitely don't want to slow any timelines. So the more that we can communicate with different people in the community, I think, one aspect is having potentially additional clinical site. In our grant application, as Rob mentioned, we have a singular Lafora site with Jose Ceratosa in Spain. It would be great if there was a US clinical site and we could get funding to support that access even more patients. And so that's something that we've already talking to other individuals that we know, clinicians or patients who have their child at a certain site, and whether the more that the doctors are interested in what our clinical study would be and would be supportive of pursuing that, we're looking to reach out to more clinicians and see if there's opportunities there. So I think it's the number one thing is let us know you're there, you're interested, and we will keep the communication lines open about opportunities.
Well, that's fantastic. Thank you. EG, I don't know who this is. Initials EG had his hand or her hand raised. Put it down, but I want to give you an opportunity to ask your question, because we've got the people online who might be able to answer. If your question was already answered, then we're good. Not getting that. So Sheila, I'm going to put you on the spot now, see what you have and what questions you have. And we're continuing to see comments on the chat from people now. Mike said any test that's needed. There's ask about I'm sorry, yeah, Mike talked about being in IT can help. What you're seeing is, in the spirit of you being such a small organization and it takes a village, it seems like our families want to provide you that village as best they can, and they don't really care whether you're a nonprofit or not. They just want to get a product to save these kids. But Sheila, what questions or comments?
Yeah, I completely understand this.
I think we answered all the ones in the chat. I've been kind of reading through and making sure. I think one of the questions, a lot of the parents on here have children that have been diagnosed several years, including myself. What can we expect from this medicine? How does it affect children that are already in the progression as opposed to non-symptomatic or in the early stages?
That's definitely a good question. I can understand. I think, I'll let Matt speak to kind of the molecular kind of findings that he's discovered in the mouse model. And I think I would just say our hope is that the mechanism destroying 4 bodies that are already present, we hope that will be, have efficacy in terms of function and recoverability. So one question is, you never know whenever there's damage or inflammation after clearing that back out, will 100% be recovered or not? And it's impossible to say until you really test it in a patient. But I think the mechanism of instead of just stopping this from continuing, we're hopefully removing kind of the toxic component that's there. And so our hope would be there would be a very significant benefit. But until you do the study, unfortunately, you don't know. But that's, that is our hope. And Matt, you can speak a little to, the metabolites and some of the stuff that he's done good at addressing. Those all suggest that improving those should have real tangible benefits. And so I think that's other great data that he's generated that we are encouraged by.
Yeah, I mean, just to kind of frame this in terms of, there's, in my opinion, 3 therapeutics that are disease altering that are in the pipeline. There's the antisense oligonucleotide, the antibody enzyme fusion, and then a small molecule to inhibit glycogen synthesis. So the antibody, the antisense oligonucleotide and the small molecule both stop disease progression. There's been a paper from the Ionis team in collaboration with Bergman Ashen's group that's been published online. And those data show quite robustly that the antisense drug stops progression in the preclinical models. And I can't overemphasize the fact that this is all preclinical data, right? This is all mouse model data. So we're just making guesses based on the mouse models. So the antisense stops progression. It's assumed that the small molecule would stop progression. Out of the three, the only that ablates and degrades those Lafora bodies is the antibody enzyme fusion. And so from the mouse data, the best guess is that that's the only drug that would completely remove the Lafora bodies and stop all of the negative cellular and brain impact that's seen with the disease. But again, as Dustin said, you don't know about reversal of symptoms until you get into the patient population. It's completely possible that the antisense oligonucleotide, just by stopping progression, you might see some reversal. We don't yet know, but the best guess would be that the antibody enzyme fusion would be the best bet to see that reversal. Yes, Nadia is asking about, has IV administration appeared effective in the Lafora mice and to what extent? Yeah, that's a great question. In the mouse model, we did an IV administration and we saw ablation of the Lafora bodies in the heart and in the muscle. In that particular study, we did not look at the brain of those mice. So we don't know if the antibody enzyme fusion crossed the blood-brain barrier. I would also say that the mice don't have a very robust epileptic phenotype, as the patients you all know have a very robust epileptic phenotype. In epilepsy, it's quite well documented that the blood-brain barrier is more allows more molecules to pass across it during epilepsy. So it's quite possible that something even of this size could cross the blood-brain barrier during epileptic activity. So the mouse model, unfortunately, is not a great model to do IV administration and then look at the brain. That said, we could do that study, and that is in discussion right now. And there's another question regarding, just to clarify, my understanding is we can't reverse the damage that's already done, like cognitive decline, speech impairment, et cetera. In my opinion, that's an unknown. You know, mouse models do not replicate that part of human physiology very well at all. That's seen for Alzheimer's models, for Parkinson's, Huntington's. You cannot predict that level of intervention from a mouse. There is, to be completely transparent and open, there is going to be in my opinion, a point of no return where the drugs don't matter. We don't know what that point is at this stage.
And Matt, if you ask anybody who has a Lafora patient in their family, particularly at certain stages of the progression, what they will report to you is an experience of good and bad. Like, it's not just a decline down, it's down and then comes back up, and down, maybe obviously a general decline, but certain days are better than others. And so I think that's what one of the things that definitely gives patients hope that maybe whatever level of whatever point where the disease is arrested, when a therapy is finally released, that they're hoping that they see not just saving their children, but an outcome that's more closely associated with those better days and weeks that they experience, obviously.
Yeah, right. And you know, I mean, in the mouse model, the valerian enabled drug performed exceedingly well and completely normalized the brain of these quite old mice. But we don't know how that would translate exactly into the human population.
We've got a few other, a lot of people on this call. You're welcome to unmute and just ask a question. But I see many of you typing in. Here's one. Nadia asked, Nadia is on our board. Would both types of administration be potentially necessary for a full body potential effect? You talked about intrathecal, you talked about IV. Could you see?
Well, yeah, I mean, in the mouse model, clearly there's Lafora, and in the patients, there's Lafora bodies throughout the body. Lafora disease is a type of glycogen storage disease, and typically wherever that aggregate is at some stage in the patient's lifetime, it causes aberrant pathology. So while I think it's the smart choice to, of course, focus on the brain initially, but if that is fixed, then there is going to be a time when you want to clear those Lafora bodies from the heart and from the skeletal muscle. And so Dustin and Rob's basket clinical trial, you know, it starts on that systemic side first because that's the quickest way to get into patients. And so not only is that the quickest way to get into patients, but it also has a potentially effective side too, because it is, as Nadia suggests, in my opinion, it's going to be necessary to clear both the brain and systemically to completely cure, if you want, to the disease.
Matt, I wonder if I could just kind of add in my own rudimentary biochem awareness. I have read a few articles, for example, on the effect of Lafora bodies on cardiovascular. And to be as delicate as I can, researchers don't seem to know because typically these patients don't tend to survive long enough to see cardiovascular effects. But it stands to reason that you possibly would once you address the more acute symptom. And so when you're talking about systemic, you are talking about all of the other organs where glycogen storage disorder could have an adverse effect. Yeah.
Correct. That's exactly right.
Okay, great. Now I'll just ask another question. Are you anticipating this a lifetime drug? I mean, I take blood pressure medicine every day probably for the rest of my life, or is this more of an acute treatment?
Yeah, in the clinical trial, you really try to figure out the right dose, how much drug is most efficacious. And then also there could be cases where you try to determine the frequency of administration and then how often. Our hope is that once you cleared out the Lafora bodies, they wouldn't progress in terms of return that quickly, and you may not need to repeat the dose that often, but that's something that would have to be determined in the clinical trial with certain endpoint markers and determination. It's not as some people may have heard of gene therapy once. This likely would be something you would repeat either every couple weeks, maybe monthly ultimately, or something of that nature. Maybe in the future, ideally, maybe every 6 months or something. But that also depends on if it's an intravenous therapy, there's the potential to treat at home just with a nurse. If it's intrathecal or ICV, that is typically then in a more clinical setting where you'd have to come in and it's a few hours infusion. And so that ultimately what's it could be different based on those different scenarios that we find ultimately in the clinical trial itself.
Fair enough. That makes perfect sense. Okay, so I'm going to pass it back to some of our other board members. I've got Kim Marino is on the line. Jennifer, I saw you earlier. Anybody can ask questions, but I'm just putting my board members on the spot. Nikki, if you have anything, you folks are living this just like the rest of them. There she is.
I do have a question. Sorry, it's 5 a.m. in the morning here in Australia. Hi everybody. I'm just about to be aired on TV, national Australian TV. So I I'm still able to ask more questions, or they've said to me, what help do I need? What can I ask them, or who can I put them in contact with, the best person to make sure that we can help?
Yeah, I mean, that's one thing we can certainly do is maybe, Frank and Rob develop a system where after this, if people have additional questions, to funnel them through a single contact point. Frank, if you want to highlight someone at your organization directly, to Rob, and we have this kind of a channel where we can, communication, time things, if there's opportunities for, to get some attention or anything we can, with press releases, anything we can coordinate, we're happy to talk with people and try to coordinate that. If there's ways to link, different, things, we're happy to try to do that. So we appreciate that. We anyone that can have a role in this, we'd like to play our parts and contribute, jointly.
Well, and that's
Yeah,
great.
I'm happy to be the point of contact for EnAble. I can put my email address in the chat. That's
Excellent.
easier. Or Frank, I think you have my email address.
Yeah, I do. And so
Okay.
why don't we talk about that? Because what I want to make sure of is that everybody gets to ask their questions and gets the information they need. I am hopeful that that you would get overwhelmed from my patients and our families. And to that point, maybe we'll talk about how best to manage that, because you are just a 2-person, now a 3-person shop. I saw that you added a research assistant. That's awesome.
Do you?
So I would love to get that information, and but just want to make sure we're not overwhelming you. And certainly, if anybody has any questions, any of the board members that are on this call, on this Zoom meeting, you can reach out to them. You can find out how to reach out to, I think all of us, our emails are listed on the website somewhere, but there's also just Chelsea's Home information at chelsea'shope.org or whatever it is. I apologize, but it's on the website. So let's, Fiza Arouj, I apologize for butchering your name, but you did raise your hand. I want to give you an opportunity to talk. If you could just go off mute and give us your question.
Hello, thank you for unmuting me. Hello everybody, this is Fiza. I'm from Pakistan. Sorry, I couldn't put on my video because it's the middle of the night. I have 2 questions actually. The first thing, do we have an age limit as to where we have the eligibility criteria for enrollment of the patients once the trial begins? Considering because of lack of exposure, by the time we identify patients with Lafora, it's already in their late teens. So that's one thing. The other thing that I wanted to ask was that you said that you've seen very good results in mice in terms of their brain that has improved. But do we mean that in terms of pathologically, or do we mean in terms of function? And if yes, then what do we mean by that?
Yes, those are very good
quickly
questions.
comment on.
Go ahead, Rob.
Yeah. Sorry, I was just going to answer the first question. A really good question. So in terms of the ages for eligibility, we don't have an exact age cutoff for the trial. It's more defined by mid-stage patients at this point. And so that's something that we're definitely going to want to answer in this trial, is if there's any variability within that mid-stage patient profile, could we then expand that beyond just, grouping mid-stage? And I'm sure you're all aware there's sort of a typical range for mid-stage in terms of ages, but we didn't set an exact hard number on that, and it'll really come down to diagnosis.
And just to make that clear, we can propose a trial, but it is a joint effort with the regulatory agency. So we could say this is what our inclusion criteria would be, but the FDA may, have other suggestions or want us to modify that. And so that's something that we can't say today, this would be approved to start a trial with a certain, our preference would be to treat as many patients and treat as early as possible. And we would, ideally try to push for that, but there are certain limitations that, we'll confront as we go through the regulatory agencies.
Okay.
I'll let Matt maybe speak to the pathology, the brain aspect.
Yeah, I mean, in the mouse models, every parameter that we've looked at that is aberrant in the Lafora disease animals is corrected with the valerian drug. So we were surprised by the metabolic profile. That's a parameter that's being used more and more as a biomarker in industry. And so it's a very strong indicator of what's going on in the brains. We were extremely excited about those data. And then more recently, we saw correction of the glycosylation defect in the Lafora disease animals after treatment with the valerian compound. And so the fact that each of those, with respect to what we can see, is completely corrected is incredibly encouraging in a preclinical stage.
Okay, thank you.
All right, let's see. Takes 3 months. So I'm just reading Mike's text chat. He's talking about, again, going back to the frequency. Yeah, there was another question. Kids asked about likelihood of the timeline. I think you spoke to that. That you're thinking end of 2022 would be an optimistic outcome. Correct me if I'm wrong on that.
We would love to start recruiting, I think, at the beginning of 2022, if we can raise funds, and the sooner the better. So we're in the process of trying to make that as quickly as possible. But again, that's with the 1221 systemically and IV therapy. The 417 is in a later stage in terms of earlier stage of development that still requires more kind of manufacturing to actually make the drug in a certain quality. So that would take longer. But for the first kind of IV systemic attempt of 1221, there's, we still hope we could begin kind of earlier 2022.
Right. Kim, you were just breaking in to present your screen. Is there something you wanted to show us?
There wasn't. I was trying to get to the chat. What I wanted to ask was, how do we get our neurologists or gene doctors more involved also?
Yeah, I think in terms of clinical sites, if there's doctors that are interested in either being a clinical site for a trial or treating patients that could be enrolled in a trial, or just better understanding what our interests are, or applying for grants, I think it would just encourage them in terms of share the links to this company, the links to this discussion, say that through Frank, we're happy to share contact information in terms of have them reach out to us. We're happy to talk to clinicians, talk them through the science, as Matt just did. We can give them details to the technology, our development plans, and we're happy to any partner that we can find, and and just encourage them to be active in the care. And just the simple fact of understanding where the patients are, what they need, that helps us not just in designing what our study is, where we estimate what patients might be out there, what is their mass, so we know if we have enough drug. Can we tell them to investors confidently that we know there's this many patients that would like drug, that we can, as we try to raise money, have a clear understanding. So I think a big step of this is trying to have the right communication and get that assembled in a way that we can benefit as we try to develop it. So I think I would just encourage everyone. I know you guys are all so kind of involved in patient care, but yeah, I encourage you to advocate for yourself, tell whatever treating physicians or doctors or whatever that you would like them to explore different treatment options and understand clinical trials and what may be inclusion criteria, and more advanced notice that we can be in communication, the better, I think is what I'd advocate for.
Dustin, I want to actually amplify what you just said. The biggest, one of the things that our families experience, and I can just tell you from my own familial experience, is that you're the only one, because it's such a rare disease. You walk down, you go to a school event, they ask about your child, you say Lafora, they've never heard of it. Nobody's ever heard of it. Some of your primary care physicians have never heard of it. I ran into earlier in our experience, which was about 8 or 9 years ago, a neurologist who had never heard of it. We never went back to that neurologist, by the way, but they feel alone. And so they want to make sure that you're aware of them. And you spoke to that. There are two real reasons why we need to know who everybody is and where they are. And one is so that they are open and they receive information about what is happening, and so that those who are doing things, such as your firm, Matt, the LECI, Dr. Saratosa has a record that he keeps in Spain. But also, you just mentioned it, you're looking for funding, you're looking for investors. Investors need to know what the market is, and that means you need to know who the patients are. You need to know how many there are and where they are. That's critically important. And Chelsea's Hope, we're working on developing a process to track this right now. And so I just encourage all of our families to reach out to us. I'm going to put the message on I'm going to share the slide that shows our website, so that you can, not just our website, but I mean our address. So if you can see it right now, you can reach out to us at info@chelseashope.org. Okay. There are 2 or 3 of us on the board who regularly see that email address and get in touch with us for anything. Tell us about your child. We will protect it, but we're not actually HIPAA qualified, so we don't have any obligations in that regard. But also, any questions you have for our researchers, please let us know. We will, often the case, we get multiple questions that are pretty much the same. And so it's the old, like when you were in school, the teacher said, hey, there's no stupid question. If you're thinking about somebody else is, well, it's the same way here. So please send us any questions, send us all the information you have. We want to make sure that we know about you, that the researchers know about you, that the drug companies know about you, and then by extension, the investors know about you, so that they can help take care of this and help our brilliant researchers provide a treatment for this horrible disease. So info@chelseashope.org. I'm going to pull that off now, but that's how you can reach us. And let's see if I have anything else. We're about 5 minutes left, so maybe we just do a little bit of a roundabout, go back to my board members, go back to Matt, Dustin, and Rob, and see if there's anything else anybody wants to hit up before we run to the end of our time.
Yeah, and again, if you think of questions later, we'd be happy to answer anything. Frank, if you get any additional questions you want to send to us, we're happy to provide later responses. And so I don't want anyone to think, as you mentioned, there's no silly questions. Everyone that's brought up things of very legitimate questions, and we want to clarify it and provide the details we can. So yeah, I just kind of reiterate, feel free to ask us questions and keep the dialogue going.
I actually see Nal just raised her hand. So now I'm going to actually break in and go ahead, please. Just go off mute and ask your question. That's great.
Hello, everyone. Thank you so much for your efforts. I have 2 kids with Lafora disease. I live in Saudi Arabia. And I think my government would pay for the treatment of my kids. But we cannot transfer them to the USA due to the circumstances of COVID-19, and due to their condition. It's progressing, and they are bedridden and with tracheostomy and G-tube. But we are willing to take the drug. We are, and we are really happy to hear about the good news. And That's it. Thank you so much. Thanks.
Thank you.
I appreciate that. And one area we just want to admit that we don't know everything, and certainly a lot of our experience, is on the U.S. and the FDA with European agency as well. But some of the parents that we've mentioned in some of these other countries, the regulatory use to be able to do the drug may be different. And frankly, I'm not going to say today we know exactly what would be the logistics of trying to make a drug available in that case. And so I think that's something that we're, in real time willing to kind of explore and consider whether, different patients could either kind of remotely qualify for a trial at a different location, or, maybe there's certain requirements that aren't needed in different instances. And so I think that's something Rob and I will discuss offline, and maybe there's certainly a way that we could kind of continue discussions to understand, every unique instance, are there different things that we could take advantage of, or try to eliminate other barriers to try to do things. And that's something we're happy to further discuss going forward.
That's great, Dustin. And that is now speaking to the international nature of this disease. And LECI, and I am multilingual in Google Translate, so LECI's asked on chat, how will selection of the study be, and when will we see compassionate use? I can go ahead and say that compassionate use does not precede a clinical trial. You have to have a clinical trial, and you have to have seen some results of that clinical trial. So compassionate use does not come before a clinical trial. Dustin, the selection criteria for the study, I think you've already, or Rob, I think you've already referenced this, but feel free to opine on it further if you like.
Yeah, maybe we can reply a little bit in writing, and just with the grant, either publish the grant application or what's listed there. But again, I would just like to highlight that it's not set in stone that this will be it. If we get different funding where we can include more patients, we will, and the FDA may have different ideas. So I think that's something that maybe we can follow up and just share what we've discussed today, at a high level, what we're currently anticipating, but then also, maybe give you some details, either a figure or slide, to say, as we look to raise money, this is how it could be modified.
That's great. And what Chelsea's hoping to do, I mean, if you have things online already, we can provide a link, or if you have electronic copies, we can host them, make sure everybody sees them. We will be posting a high-level summary of this event, and as I mentioned, it is being recorded, so people can go back and view it later. At this point, we are at 12:31. I want to respect everybody's time. Again, we're going to have to cut it for now, but that doesn't mean that the opportunity for anybody to ask questions and get answers is cut. Reach out to us at info@chelseashope.org. About to flood Lena's inbox, but I hope I am. And we will get the recording of this accessible for everybody, as well as a high-level summary, a written summary of it. So Rob, Dustin, Matt, my board members, and mostly important, all of our families, thank you very much. Truly appreciate it. It's just, it's incredible to be able to have such direct access to the people who are trying to save our children's lives. So thank you very much.
Again, thank you, everyone. And no one's alone in this. And we definitely, appreciate everyone's effort. And Frank, we really appreciate you putting this together. We just want to do our part.
Stand up here and look like I look.
Okay. Thank you so much.
Thank you all.
Thank you very much.
Bye-bye.
Take care.
Thank you.
Bye, everybody.
Bye.
Thank you. Bye.